I need this and this on. Okay, so we're going to do things really quick. I took one of the videos off because it was really long and annoying. So we just have one video to watch today, and then I need you to go into the Are You a Night Owl, Morning Lark, and for the pre-lab, you're going to answer the questions at the very end of the lab to see if you're a morning person or a night person, and then we're going to do the genetics behind it to see what you are. Okay, so should be fairly easy.
Pretty easy, so then we have time to work, and then next time lab and stuff.
And the lab next time should be fairly easy as long as you've read through it and know what to do and have written it up in your lab notebook.
I am not a morning lark or a night owl.
Okay, so I'm going to go right to the videos first. And we're going to get started on 13. And it's not very long. I'm actually going to put it at 1.5 speed.
I'm a permanently exhausted pigeon.
Yes, this is one note that you do need to put notes in.
Yeah, 1 second.
Wait, where is it?
How can you chill?
I'm just fixing this up, and then I'll go back.
I'm a permanently exhausted pigeon.
I don't have the video. Why does it not?
The billions of people. Imagine you were alive back in the 1980s and were told that computers would soon take over everything, from shopping to dating and the stock market. The billions of people would be connected via a kind of web, that you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality, and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. Where it came from, what we're doing right now, and about a recent breakthrough that will change how we live and what we perceive as normal forever.
It's still 1 1/4 inches.
Humans have been engineering life for thousands of years. Through selective breeding, we strengthened useful traits in plants and animals. We became very good at this, but never truly understood how it worked, until we discovered the code of life, deoxyribonucleic acid, DNA, a complex molecule that guides the growth, development, function, and reproduction of everything alive. Information is encoded in the structure of the molecule. 4 nucleotides are paired and make up a code that carries instructions. Change the instructions, and you change the being carrying it. As soon as DNA was discovered, people tried to tinker with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to get a useful plant variation by pure chance. Sometimes it actually worked, too. In the 70s, scientists inserted DNA snippets into bacteria, plants, and animals to study and modify them, for research, medicine, agriculture, and for fun. The earliest genetically modified animal was born in 1974, making mice a standard tool for research, saving millions of lives. In the 80s, we got commercial. The first patent was given for a microbe engineered to absorb oil. Today, we produce many chemicals by means of engineered life, like life-saving clotting factors, growth hormones, and insulin, all things we had to harvest from the organs of animals before that. The first food modified in the lab went on sale in 1994, the Flavor Saver tomato, a tomato given a much longer shelf life via an extra gene that suppressed the buildup of a rotting enzyme. But GM food and the controversy surrounding them deserve a video of their own. In the 1990s, there was also a brief foray into human engineering. To treat maternal infertility, babies were made that carried genetic information from 3 humans, making them the first humans ever to have 3 genetic parents. Today, there are surrounding them deserve a video of their own. In the 1990s, there was also a brief foray into human engineering. To treat maternal infertility, babies were made that carried genetic information from 3 humans, making them the first humans ever to have 3 genetic parents. Today, there are super-muscled pigs, fast-growing salmon, featherless chickens, and see-through frogs. On the fun side, we made things glow in the dark. Fluorescent zebrafish are available for as little as $10. All of this is already very impressive, but until recently, gene editing was extremely expensive, complicated, and took a long time to do. This has now changed with a revolutionary new technology now entering the stage, CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work? Bacteria and viruses have been fighting since the dawn of life. So-called bacteriophages, or phages, hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell, microorganisms, plants, animals or humans. But despite the revolution CRISPR is for science, it's still just a first-generation tool. More precise tools are already being created and used as we speak. In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients in the lab, proving that it was possible. Only about a year later, they carried out a larger-scale project with rats that had the HIV virus in basically all of their body cells. By simply injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from cells all over the body. In a few decades, a CRISPR therapy might cure HIV. And other retroviruses, viruses that hide inside human DNA like herpes, could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever. But all of these medical applications have one thing in common. They are limited to the individual and die with them, except if you use them on reproductive cells or very early embryos. But CRISPR can and probably will be used for much more. The creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene pool. The means to edit the genome of a human embryo already exist, though the technology is still in its early stages, but it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore. Early on, vanity traits will mostly be left alone. But as genetic modification becomes more accepted and our knowledge of our genetic code enhances, the temptation will grow. If you make your offspring immune to Alzheimer, why not also give them an enhanced metabolism? Why not throw in perfect eyesight? How about height or muscular structure? Full hair? How about giving your child the gift of extraordinary intelligence? Huge changes are made as a result of the personal decisions of millions of individuals that accumulate. This is a slippery slope. Modified humans could become the new standard. But as engineering becomes more normal and our knowledge improves, we could solve the single biggest mortality risk factor, aging. 2/3 of the 150,000 people who die today will die of age-related causes. Currently, we think aging is caused by the accumulation of damage to our cells, like DNA breaks and the systems responsible for fixing those wearing off over time. But there are also genes that directly affect aging. A combination of genetic engineering and other therapy could stop or slow down aging, maybe even reverse it. We know from nature that there are animals immune to aging. Maybe we could even borrow a few genes for ourselves. Some scientists even think biological aging could be something that eventually just stops being a thing. We would still die at some point, but instead of doing so in hospitals at age 90, we might be able to spend a few thousand years with our loved ones. Research into this is in its infancy. And many scientists are rightly skeptical about the end of aging. The challenges are enormous, and maybe it is unachievable. But it is conceivable that people alive today might be the first to profit from effective anti-aging therapy. All we might need is for someone to convince a smart billionaire to make it their next problem to solve. On a bigger scale, we certainly could solve many problems by having a modified population. Engineered humans might be better equipped to cope with high-energy food, eliminating many diseases of civilization like obesity. In possession of a modified immune system, with a library of potential threats, we might become immune to most diseases that haunt us today. Even further into the future, we could engineer humans to be equipped for extended space travel and to cope with different conditions on other planets, which would be extremely helpful in keeping us alive in our hostile universe. Still, a few major challenges await us, some technological, some ethical. Many of you watching will feel uncomfortable and fear that we will create a world in which we will reject non-perfect humans and pre-select features and qualities based on our idea of what's healthy. The thing is, we are already living in this world. Tests for dozens of genetic diseases or complications have become standard for pregnant women in much of the world. Often the mere suspicion of a genetic defect can lead to the end of a pregnancy. Take Down syndrome, for example, one of the most common genetic defects. In Europe, about 92% of all pregnancies where it's detected are terminated. The decision to terminate a pregnancy is incredibly personal, but it's important to acknowledge the reality that we are pre-selecting humans based on medical conditions. There is also no use in pretending this will change, so we have to act carefully and respectfully as we advance the technology and can make more and more selections. But none of this will happen soon. As powerful as CRISPR is, and it is, it's not infallible yet. Wrong edits still happen, as well as unknown errors that could occur anywhere in the DNA and might go unnoticed. The gene edit might achieve the desired result, disabling a disease, but also might accidentally trigger unwanted changes. We just don't know enough yet about the complex interplay of our genes to avoid unpredictable consequences. Working on accuracy and monitoring methods is a major concern as the first human trials begin. And since we've discussed a possible positive future, there are darker visions too. Imagine what a state like North Korea could do if they embraced genetic engineering. Could a state cement its rule forever by forcing gene editing on their subjects? What would stop a totalitarian regime from engineering an army of modified super soldiers? It is doable in theory. Scenarios like this one are far, far off into the future, if they ever become possible at all. But the basic proof of concept for genetic engineering like this already exists today. The technology really is that powerful. While this might be a tempting reason to ban genetic editing and related research, that would certainly be a mistake. Banning human genetic engineering would only lead to the science wandering off to a place with jurisdiction and rules that we're uncomfortable with. Only by participating can we make sure that further research is guided by caution, reason, oversight, and transparency. Do you feel uncomfortable now? Most of us have something wrong with them. In the future that lies ahead of us, would we have been allowed to exist? The technology is certainly a bit scary, but we have a lot to gain, and genetic engineering might just be a step in the natural evolution of intelligent species in the universe. We might end disease. We could extend our life expectancy by centuries and travel to the stars. There's no need to think small when it comes to this topic. Whatever your opinion on genetic engineering, the future is approaching no matter what. What has been insane science fiction is about to become our new reality, a reality full of opportunities and challenges. Videos like this would not be possible without viewer donations on patreon.com.
Okay, so what we're going to do now, hopefully you've got a computer in front of you. I would like you to go there. I've set the link up here. This needs to be set up in your lab notebook. It can't be all done online. It needs to be in your lab notebook, and you're going to submit it in your lab notebook. So you're going to go to, are you a morning lark or a night owl? And then if you scroll down, it basically gives you an overall view. The synopsis is what overall are we going to do here. Background gives you why it's significant. Excuse me. And then it says let's get started. This is where we basically, this is where we basically start the lab, and it'll tell you what we need to do. We're going to be doing a cheek cell extraction, PCR setup, and then we're going to be doing electrophoresis at the end. And it tells you how to do it. And at the very end, if you come down here, running the gel, what you're expected, it has a questionnaire. I'd like you to use this questionnaire as your pre-lab. So you go through, put your name on there, do all what you answer your questions. Then at the very end of it, it'll tell you what your score is, kind of thing, and when your onset wake up kind of thing is. So I want you to sort of do a pre-lab and a hypothesis of what you're going to see in your genes by how you react. Yes?
And then, do we need to write the questions or just write our answers to the questions?
For the student guide, the questionnaire, I would just say, so like on number 1, approximately what time do you get? I'd get up at blank, and then put how much is worth. So summarize, like you're rewriting it as the, does that make sense? You don't have to write the whole thing. So approximately what time do you get up? I get up at, is what I would write. I get up at blank. Approximately what time do you go to bed? I go to bed at blank. And I would write the number by it so that I could add them up after. Does that make sense? Okay. Any questions? Okay. So all your work is due March 17th. So if you're behind, you need to get your work in by March 17th.
7 days.
That's
Yep, 7 days.
That's nothing.
Wait, how up to what assignment do we have to have by then? We don't have to have 12, right? Because we're only on like 11.
No. 12, what do I have on? 12 will be due on the 20th.
So we have to have up to 11.4.
Yeah, well, 11.4 is due on the 18th. So 11.3 by the 17th, and then these two last ones. Yes, the quiz for today needs to come in and be done, yes. The questionnaire goes in your lab notebook, yes. So that's your pre-lab in your lab notebook. If you need a lab notebook, I have some over on the far wall over by the howling wolf. If you have lost yours or you need a new one. Okay? Time is yours.